Hi everyone, Amy and Annalie back here to talk about the plasma membrane and transport in and out of the cell across this membrane. If we start here with this picture just as a reminder of what the plasma membrane or the phospholipid bilayer looks like, remember that we have two layers of phospholipids that have a polar head group. Here and here is polar. And the middle, that is non-polar. I know we keep saying that, we keep reiterating that, but it's really important. So let's remember, two phospholipids will form this bilayer just like this spontaneously because they are all trying to keep the water out. That means that the tails are hydrophobic. They hate water, or they are afraid of the water. The heads are hydrophilic. They love the water. They will interact with the water just fine. And so the heads face out towards the extracellular fluid or towards the intracellular fluid, also known as the cytoplasm. So, funny handwriting there. Okay. Now, if you also take a look at this membrane, just to show you, we have lots of proteins that are here in blue. These proteins and some sugars even that are attached, these can act as markers just to identify a cell as one type of cell or another, or these proteins can act as transport molecules that actually help decide what is able to move in and out of the cell membrane. If you'll notice here, you also have cholesterol molecules here in yellow, and these are actually a major part of our cell structure. So don't think that cholesterol is all a bad thing. The cholesterol is actually a really important piece of our plasma membrane. All right, let's move ahead. When we talk about transport of molecules in and out of our cells, if this is our membrane, right, we have things going in and out all the time. But our plasma membrane is referred to as selectively permeable. That means that some things are allowed in and out and some things simply are not. That'll be based on quite a few different characteristics of whatever the molecule is. But this membrane is going to decide what's able to move in and out of the cell. Now, before we talk too much about that, we need to discuss a few ideas about these molecules in general. One is that all the molecules we're going to be talking about have what's called kinetic energy. Now, that's a bad star. Wow, let me try over here. Kinetic energy is the energy of motion. So if you think about everything that we're discussing here in our body, these molecules are constantly moving. They're basically bouncing around, and they're going to bounce around and bump into each other and bump into other structures. You can see here in this picture, here's a molecule. It's going to bounce that way. It's going to hit the wall of the box and bounce this way. It's going to bounce into another molecule and ricochet the other way. So there's constant moving happening with all these molecules in our body. Now, when you put molecules together, if you have what we would call a high concentration in one area where you have a lot of things clumped tightly together, and a low concentration in another area with only a few molecules with lots of space in between them, what's going to be the natural tendency? As all these molecules bounce against each other, these on the left-hand side are going to bounce and ricochet off of each other quicker than the ones over here. And because of that, that's going to send them farther away. Well, what happens is that through the kinetic energy, or this constant motion, the molecules are going to bounce around and bounce against each other in such a way that they will eventually fill this container evenly, where there is an equal distribution of molecules. We call that moving down the concentration gradient. That means they go from the high concentration on this side to the low concentration on this side until there is an equal distribution within the entire space. Let me show you another picture of that. Here's another idea. In this example, you can see that we're basically using color to show the high concentration, this dark black region, shows the high concentration here on the left, and a low concentration on the right where you see the lighter color. Movement is going to occur to the right. These molecules are going to move from the area of high concentration to the area of low concentration until you have a completely even distribution of molecules across that space. Again, we say that we are moving down a concentration gradient when this is happening. We're going from high to low until we reach what's called equilibrium or balance. Now, we talked about equilibrium when we talked about negative feedback, that equilibrium was going to be that state of balance, the normal state. When we're talking about equilibrium in this case, equilibrium means an equal number of particles on one side versus the other, or an even distribution. Okay? This is one other aspect of this concentration gradient that has to do with how fast molecules move from one side to the other. The steeper the gradient, meaning the more molecules you have over here versus over here, the more molecules you have on one side versus the other, the faster that movement is going to occur. Okay? It's going to go faster here in the figure at the top because the concentration gradient, the difference between the two sides, is a lot greater than here, where the amount or the concentration of molecules on one side versus the other is a little bit closer together. So the steeper the concentration gradient, the faster this movement is going to occur. All right, when we discuss membrane transport, we need to group this into a couple of different categories. The first type of transport that we're going to talk about is passive transport. Passive, just like you would think about, if somebody is being really passive, even in terms of their personality, they're not doing anything really forceful. They're being pretty laid back. They're going to be pretty natural, potentially. I'm not talking about passive-aggressive, just being passive, right? You're going to let things happen. Well, passive transport means really just that. It means that there's no energy. Remember, energy is ATP. There's no energy input required. The movement of these molecules is going to happen simply on their own, not having to make anything happen. It's really going to go back to the idea of all that kinetic energy that's causing these molecules to bounce around and ricochet off of each other, that these molecules are going to naturally have the tendency to move in this direction. So this type of passive transport, again, is going to be movement from a high concentration to a low concentration. It happens naturally, no ATP. It is simply the bouncing around of these molecules eventually leads to movement from the left to the right in this example. Or it might mean the movement of molecules from the outside to the inside of the cell, or from the inside to the outside. But with passive transport, there is no ATP input required. It's just a natural movement that's going to occur. Let's talk about a few types of passive transport. The first type of passive transport is called simple diffusion. So diffusion, let me define that really quick. Diffusion is the movement of a molecule sometimes called a solute molecule. Solute molecule means something that's dissolved in water. We need to specify solute because in a little bit we'll talk about the movement of water molecules themselves. That's something totally different. But the movement of a solute is referred to as diffusion. And this is going to be the movement of a solute through a membrane. Okay, let me clear all of this out so we can keep talking about this. So simple diffusion is the movement of a solute molecule directly through the membrane. No transport proteins required. That's important because there are some molecules that are able to simply move through this membrane without needing any assistance at all. It will naturally happen. You can't really stop it. They are somehow drawn through this membrane, and it will happen based on that rule of concentration gradient. So these molecules will always move down their concentration gradient from a high concentration to a low concentration. Now, there are only particular types of molecules that will fall into this category of simple diffusion, and these are going to be molecules that are small enough to actually fit through the phospholipids, and it's going to have to do with their charge. We have polar molecules and we have nonpolar molecules. Which type of molecule do you think is going to be able to interact with this internal portion of the plasma membrane? Polar or nonpolar? Well, the types of molecules that can go directly through the plasma membrane are the nonpolar molecules. Particularly things like oxygen and carbon dioxide. Those are both small, nonpolar molecules that are able to simply fit directly through that plasma membrane without needing any assistance. If you were to think about a charged molecule, let's say that this one here had some charges on it. If this molecule tried to go through the plasma membrane, it would get repelled. Remember our discussion about oil and water? A charged particle is not going to be able to go through the nonpolar interior of the plasma membrane at all. So this charged particle would get bounced out. So the only thing that can go through happily and without any interference would be small nonpolar molecules, particularly like oxygen and carbon dioxide. Now let's talk about some other examples. The other type of passive transport we're going to talk about is facilitated diffusion. Now, because this is diffusion, we are still talking about the movement of a solute molecule through the membrane. But because it has the word facilitated, we know that this time it's going to require the help of some type of transport molecule to get through. So our idea of the small nonpolar molecules that we're able to go directly through, remember? Those are really handy, but they have to fit into that category of being small or nonpolar. So molecules that don't fit into those two categories are going to need some type of help. So let's talk about what those would look like. Let's see. So two different types of facilitated diffusion. One is going to be channel-mediated. Channel-mediated diffusion is what you see here in this example. This is an open channel where fluid and molecules are able to go through. I like to think about this almost like a trash can. Sometimes they have a lid, sometimes they don't in channel-mediated diffusion. But these molecules usually are small and charged, so they are polar, like a sodium ion for an example. It would go through this channel from one side to the other. Now, what type of characteristics do you think would have to be in place for this channel? Well, the charged particle is having to avoid this internal nonpolar interior of the membrane. So what this channel does is that it shields the interior, or it shields this molecule from all that nonpolar area inside the membrane. So this allows ions like sodium, chloride, potassium. You see these charges, these electrical charges that would normally get bounced out of the membrane itself? They're able to pass right through because they're being shielded by this channel protein. And that's a pretty handy thing because we need all these ions to flow through on a regular basis to help us with membrane transport, to help us with muscle, to help us with nerve transmission, all of that.